We'll see. Let's see how you did on this one. Distinguish between auto and heterotroph. Somebody had asked that. So a cell feeder versus something that's got to eat something else. You know, live off of something else, right? So distinguish between ectoparasite, endo, and intracellular. Was that a fair question? So ecto lives on you, endo lives within, but intracellular is inside the cells. So fair. And a parasite is just something that's going to live off of a host cell at the expense of a host cell. What's meant by an essential nutrient is something that your cells can't make. You've got to bring it in. You can't make it. Your living cells are pretty amazing at what they can build as building material. Like they can take monomer units and build what they need, but there's some things they can't. So that's what's referred to as essential. That was fair, wasn't it? I think I went over that. True or false? Trace elements are those needed in such small quantities that they don't contribute significantly to the health of an organism. That's false. They're trace elements, but they are absolutely needed, you know, for the health of the organism. Did that seem like a trick question? It didn't need to be. Yes?
Wasn't that initial statement of the question like it is a trace element and then it's not?
Trace elements are those needed in such small quantities that they do not contribute significantly to the health of an organism. Not true. They're necessary. They're necessary for the health. So, you know, I think a lot of time is spent on macronutrients. It is true that a lot of information is given about carbs, proteins, nucleic, you know, and all of that stuff. But we do need those trace elements. So, I thought I had actually said that out loud. Had I? Some people are saying that. Okay, so the ability of a fluid to affect the cell size, I gave you this right before class, is tonicity, is what you're talking about, is the fluid's tonicity. Describe the environments needed for an aerobe. They need oxygen. They need oxygen. Anaerobe?
No.
Oxygen's toxic, essentially. You can't grow in the presence of oxygen. And then I don't know, I'm going to admit this next one, but what's a capnophile?
It needs a lot of CO2.
It needs CO2. Yeah. Okay, so it needs CO2. Campylobacter jejuni is in what kingdom? It's a bacterium. It's bacteria that has been linked to a rare sequelae known as Guillain-Barré syndrome. What is the significance in understanding the different growth phases of microorganisms? How is this useful? And I sort of in the last, I might omit this, but in the last few minutes of the class, I talked about how in the beginning phase, when you're introduced to a potential pathogen, there's going to be this kind of lag time, but then there will be this logarithmic exponential growth. And that's when they're the most, that's when you're going to have the best chance of treating them there. So that's what I meant. But how many, do y'all feel good about that question? Exponential phase. What is meant by the description of signs and symptoms of disease as being subclinical? That you cannot even, you just do not even know how long it took me to phrase that sentence. I was actually worried about my mental health. I was like, oh, I asked that. Okay, y'all there. It means what? Under the radar. Like it's subclinical. You're not really seeing signs and symptoms. I remember Janet talking about it in class though, and answering it. And so hopefully you guys had that. You understood what subclinical means, that you're really not seeing the signs and symptoms. And that's what we need for presumptive diagnosis, right? The thing that will make you the craziest, and you all know people who do this, are the people who think, I'm going to go in and I'm just going to see what they find out about me. I'm not telling them anything. You know, how many of you know people who go to the doctors and actually do that? So you know that. But the fact is, it's so important that practitioners get as much history as possible and that patients tell them as much as possible, because that's where the presumptive diagnosis comes from. And from that presumptive diagnosis, you can figure out what testing you do need to do to confirm a definitive one. But I just think it's amazing that still in 2019, there are people who think, well, they'll know what to do. No, they won't. You have to tell signs and symptoms. So, okay. And you've just got to really encourage, I was going to say students, but I meant patients, to do that. Okay, when a person's immune system produces specifically shaped proteins known as antibodies to fight against a particular foreign antigen, and then those antibodies start to interfere with the person's own cells, what kind of disease is that? Did everybody get that? Like, I think you all need 100s if you got that. Did you get that right?
I mean, if you want to do all 100, like, I won't complain.
But you had a bonus. So what is Ebola? Ebola is a virus. So you needed to say it's a virus, and if you knew that it causes hemorrhagic disease where you start bleeding out, and that's fine too. But I really just wanted you to know it's a virus. At this point, and I think we talked about it, you should know that it does cause a hemorrhagic disease that has an incredibly high mortality rate, doesn't it? All right. So which one should I throw out? So should I throw some out?
I'm sure I'll get 100. Taking it.
The parasite one?
No breathing. I knew Ebola was a virus, but because I had it in my notes, obligate parasite.
Okay. Well, viruses are obligate parasites. You know, I don't know, guys. You know, I'm nicer than y'all think I am. Y'all think I'm so mean. All right. So I had ended with this kind of, and I probably had jumped to it, but I had ended with the idea of that when microbes are in that exponential phase, that's when they're the most vulnerable to treatment. So that what we hope for is that we're getting good signs and symptoms that we can get to diagnosis and know what we're treating, so we can hit it during that phase. Yes. That's great. That's another way to say it. So a lot of different ways to say that, and that's exactly right. And the only thing I kind of skipped by in this, but I think, you know, you guys should have an idea. Not all microorganisms have the same division time. So this can also help us to explain why some microbes, the progression of the disease can sometimes be sort of slow onset. And then because the microbes are taking a little time to do that, but also an idea of a clinical microbiology lab, which we said the hardest thing is that we've got to get the answer to this as quickly as possible. But first, we've got to get the microbes. Sometimes we've got to get them growing out, and some of them are really slow. One of the ones I wanted you to know is so slow is actually Mycobacterium tuberculosis, and leprae too, but tuberculosis. And this is to grow. It can take weeks to get this actually growing in a lab. I just saw on a social media site, Facebook. I'm not sure why I look at it anymore, but anyway, somebody had put up from a site that I guess they think is legitimate, a picture, because these sites are made to look legitimate, a picture of somebody that was supposedly an illegal immigrant crossing the border, and it was showing this person as having cutaneous tuberculosis. And so this supposed news site was saying how this is an untreatable disease, and there's no vaccine available in an untreatable disease. Is any of that true? Absolutely not. Absolutely. Like there was hardly any facts in that statement. So, this is what's coming across your southern borders. Absolutely not true. There is an immunization against tuberculosis. It's just the United States has not chosen to do the immunization because we have such low numbers of that disease in our country. And I do want you all to understand this idea about this. There is a vaccine against tuberculosis, and most developed countries use it. So most developed countries' citizens have had an immunization against tuberculosis, which you could say you'd be really happy about because the numbers are really low because there have been immunizations. So why isn't the United States doing immunizations against tuberculosis? I'm not sure why I brought this up right now. Did that say something? What'd you say?
Because we have so low numbers.
Because we have low numbers, but that's not why. Because we have low numbers and we want to do that immunization too. The reason the United States Health Services have decided not to immunize, which this site said there isn't one, and do you know how many times it was shared? Okay, we're not going to talk about that. But okay, the reason the United States has not is because a couple of reasons, but it's because we use the rapid skin test to diagnose the disease. So we use a little, how many of you had the PPD test? How many of you had the skin? And now we have a blood test too that can do it. But what those are indirect serology tests. Would you tell me an indirect serology test is testing for? An antibody. So because we can take indirect serology tests and test our health professionals, have them test once a year, we can see, Abby, if you've been exposed to TB. And if your test is negative, do you have TB? You don't have TB, you haven't been exposed to TB, keep on working with your patients and using universal precautions, you know, whatever. So we can use those tests as diagnostic tests in our population. If we start an immunization process in our country, can we use those tests anymore for diagnosis? No. No, because if we immunize somebody, would they always look positive? Yes. They would always appear positive. So we have made the decision in this country, and we're one of the few developed countries that have done this, but we made the decision to use the rapid indirect serology testing, whether it's the PPD or whether it's the blood test. We made that decision to use that for diagnostic purposes, where if we try to start a mass campaign of immunization, we would never be able to do that again. And what we would have to do is get a direct serology being positive. And by the way, we can't find those for TB. We would have to get it growing in the lab. And what did I just tell you about the microbiology lab and the handicap we have in that lab about mycobacterium tuberculosis? It takes weeks to get the diagnosis back. And by that time, if that person has truly been coughing and hacking up TB, have they spread it potentially? Do you get what I'm saying? So much was wrong. I mean, everything was wrong about that thing, but it was being shared. Conspiracy theories. Okay. So anyway, so when you see something like that, please bring it to people's attention and try to explain to people when there's inaccuracies and fear-mongering and, you know, whatever about those kind of things, because that's all it is. It's actually from uneducated, okay, and or people having an agenda with sites. But anyway, I don't know why I brought that up. What did that have to do with this? Making
because like the generation that on that grow.
Oh, the generation times. Right, it takes a long time to grow those out, and it would take a really long time. And there are no direct serology tests because somebody can have like tuberculosis and active tuberculosis, but to be able to get to that pathogen, you have to go in and do like a lung biopsy. And most people aren't going to let you do that, are they, to do a direct serology test? So what we can do now, though, we can do the indirect because we don't do immunizations by choice. We can use the indirect serology test for diagnosis, and then we back it up with lung x-rays, and then we can do actually prophylactic treatments. If somebody has one of those as being positive, they'll go on prophylactic treatment for 6 months. If somebody doesn't have active TB, which means that they're actually in their sputum cultures, they're active microorganisms, then they're not contagious. It's not as contagious as people think it is. They have to have active sputum cultures. Once somebody has 3 negative sputum cultures, they can come out of isolation. That's just protocol. So, you know, well, whatever. It's just being smart about it. You don't have to just do it. But we do talk about also like quantification. Quantification means that you know numbers of things. And I've already introduced you all to the idea that we live and are in a microbial world. They live in us, they live on us, they're allowing us to live in a state of health and well-being, but we can use their numbers sometimes to figure out whether they are opportunistic pathogens or not. So we definitely have ways that we can actually evaluate how many are present. So that's used in helping to determine when we're talking about an opportunistic pathogen. Now there's some microbes we said should never be there in any numbers. And those were the true pathogens, and is tuberculosis one of those. Yeah. Okay. So what I want to do and what I need to do is, you guys were skipping chapter 7 because I just am. I don't think there's actually a few chapters probably should that I'm going to skip a little bit. I think I'm going to skip, but anyway, chapter 7 is one of them. So we're skipping that. But we're going on to chapter 8, and most of you guys have actually had this. So you have a strong background in this. How many of you feel really good about genetics because you had me for MP1? So raise your hands if you had me for MP1 because you better feel good about it. This is fun. Thank you, Janet. So this should be a review, really. Most of this should be a review for you, but I do understand that some of you haven't, had that. So I only, in this class, only test you on what we talk about. So as we're talking about these topics, even though I do think it's a review for a lot of people, if you have any questions, stop me as we go. Fair enough? So chapter 8 is dealing with genomes. And so when we think about this, in genetics, and so when we think about this, we know that this is how we're looking at the DNA, aren't we? So we're looking at the DNA and how DNA is actually transferred and the transfer of those traits from parent to offspring. So genetics is study of inheritance and anything living. This is what we're going to look at. And again, this genome means the sum or the total of the genes, if you will, for a particular organism. We already know that bacteria have chromosomes. And what do those chromosomes look like? And this will be down here. What does a bacterial chromosome look like? Did we talk about? Okay, so bacteria have a single chromosome, good, Janet, a single circular chromosome, right? So that's what bacterium have. How about viruses? What did we say about viruses? They can either be what or what? They're either going to be DNA or they're going to be RNA, depending on the viral agent. And then we know some species are either asexual or sexually reproducing species. So when we were talking about eukaryotic organisms, which include which kingdoms? At this time, y'all should be really fluent about eukaryotic versus prokaryotic is bacteria, right? Prokaryotic means bacteria or archaea. Eukaryotic are those 4 kingdoms. They're what? Plants. So that's exactly right. So those, and we know in those 4 kingdoms you can either have asexual reproduction, where you just have a parent that is able or capable to actually have their DNA replicate itself, and the one parent cell divides into 2 parent cells. And in asexual reproduction, that's a type of cloning, isn't it? Yeah. And in some eukaryotic species that, way more complex species, you actually have sexual reproduction, which means that the offspring are going to get half and half their genetic information from each parent. Fair enough. Okay. So we're going to talk more about it. And some of this we've had, but that means some of this we've even talked about in here. So, all right. So I'll get, if I get to something I really want to write down, I probably should. So there's different types of genes. When you hear the word gene, and I want you to know when you hear the word gene, what you're really talking about is this recipe for a particular trait. And another word that you could use for trait, what do my people know? What's another? A characteristic or a trait, a characteristic recipe for a protein to be made, right? So some are structural, which means that they're coding for proteins. And some are regulatory, which means they're regulating. They're going to be regulating the gene expression. So there's different classes of genes, but we know kind of what that means. Now, when you talk about the terms phenotype and genotype, I think you all mostly remember that phenotype is what is expressed, what you can see. Is that right? So it's what's expressed. And genotype means that you know all of the actual nucleic acid information that's present. It means you know everything that's present. So sometimes we talk about that you know from a certain phenotype what the genotype must be. When is that the case? What phenotype do you know if you wouldn't know the genotype? Would you say? Phenotype, so when we talk about phenotype, it means what's expressed. It means whether you have that trait or whether you don't. Is that right? That's phenotype. Whether this trait is being expressed or whether it's not. So we talk about that as being dominant versus recessive, correct? So dominant simply means that you have the recipe for a trait to be expressed and it will be expressed. Phenotype, recessive meant that you don't have the information. If you don't have the information, can you do it? No, and neither can those cells. If they don't have the information, which means the gene, which means the recipe for the trait, they can't make it. So the phenotype is recessive. And so would you know the genotype? Yeah, absolutely. You wouldn't know that there was no information there. Because if it was there even once on a sexually reproducing organism, wouldn't it be expressed? Wouldn't it? It would be expressed. So simply, I'm trying to say it in different ways, but phenotype means, is it dominant or recessive? Is it producing this? Is it not producing this? But genotype means you know exactly at a molecular level exactly what's on the chromosome. If something's recessive phenotypically, you know the genotype. But if it's a sexually reproducing organism and it's dominant, you don't know the genotype because they could be heterozygous or they could be homozygous dominant. So does that make sense?
Recessive has no information.
Well, recessive means they're not producing that particular protein, so they're not producing that recipe. Okay, so let me think what else I wanted to tell you about this. So when we talk about genes and when we talked about expression of traits, we have actually talked about chromosomes, and we talked about this little circular chromosome that bacteria had, and we talked about individual chromosomes that eukaryotes have. Let's give an example for humans. How many chromosomes do humans have typically? So 23 pairs of chromosomes. We call it 46 total should be, but 23 pairs. Why do we think of our chromosomes in pairs? Because we get one of the homologs from our one parent and we get the other matching one from our other parent. When I say matching, I don't mean that they have the same information for traits. I just mean that it's the partnered homolog for that pair, right? So does everybody remember that depending on the eukaryote and the sexually reproducing organism, they would have different numbers of chromosomes, but they're always expressed in pairs. You know, plants are sexually reproducing, fungi can be sexually reproducing, animals are sexually reproducing. So we think about those chromosomes in pairs, all right? Where, and bacteria is a circular, one circular chromosome. But do y'all remember me also talking to you about plasmids? And I said plasmids are going to be these little circular pieces, little extra pieces of nucleic acids that can hold a trait, hold information for traits. So these are plasmids, and we have talked about those. And then we looked at viruses, and we see viruses may be have either a strand of DNA or a strand of RNA, right? Now,
They can carry plasmids have extra pieces of what?
Plasmids are these extra little pieces of nucleic acids that can hold traits. And if the cell has those plasmids, would they be being expressed? If there was recipe for something, would it be expressed? It would be. It would be expressed. Now you kind of have to know this going forward here. So any questions about this? Okay. All right. Okay. So when we talk about DNA, and I know this is review for most of you, but when we talk about this molecule called DNA, we said that you would think that this molecule must be incredibly complicated. You would think that it must be so complicated when you think even within one species, Homo sapiens, in room 65 at the SVCC Daniel campus, you look at just one species that we can see, we've got a lot of other species that are microscopic, but just our species, Homo sapiens, you look in this room, is there a lot of diversity in this room?
Yes.
There's diversity in height, there's a diversity in body structure, there's diversity in eye color and skin tone, and it, you know, there's so much diversity in this room, and you're telling me that all of that diversity is because of the DNA molecule? Right? That's what you're saying. So we do understand that this DNA molecule, we would think with all the diversity you see with even in one species, that the DNA molecule must be incredibly complex, and it must be so different between species. Think about just insects. There's more than 1 million different, I can't say that word, million different insects. And all of the differences that you see just within the insects is because of their DNA. So you think, okay, this has got to be complicated molecule, right? It must be incredibly complicated. And then we go down to like viruses and bacteria. That's just got to be so different as far as the structure goes. But what we know is that actually this molecule is not so complicated. And the structure of this molecule is the same between all life on this planet, even that things that we call non-life, which are those viruses that we say are active or inactive, but not really living because they can't carry out any of the metabolism needed for life processes. They must be inside of a host cell and take over that machinery. So it's crazy to think that this molecule is actually as simple as it really is. And I do want you to remember that the basic structure of this DNA, whether we're talking about this DNA being a circle, like we find in what? In prokaryotes, which are bacteria and archaea, or whether it's in little pieces, little snippets called plasmids, or whether it's what? In chromosomes, which what we think of more complex organisms as having, right? So no matter what, this is the structure. And what we know is that it's double-stranded, and you would want to remember it's double-stranded. You would want to know that we think about it as a ladder, don't we? With rungs in the ladder and the strands being the sides of the ladder. Everybody good with that? And we understand that these sides of the ladder couldn't be more boring if they tried, because what they are simply phosphate, a sugar, which is deoxyribose, phosphate, another deoxyribose, phosphate, guess what's here? Deoxyribose, guess what's here? You get what I mean? It's just repeating. Phosphate, deoxyribose, phosphate, deoxyribose, phosphate, deoxyribose. That's the strands, right? But then there's these rungs in the ladder. And when we look at the rungs, we would expect there to be, this is where the complexity comes. This is what leads to all the diversity, right? Except that no, it's not so complex either. There are 4 nitrogenous bases. There are 4 bases that are found in DNA. And those 4 bases are adenine, thymine, cytosine, and guanine. They're the bases. You mean that's all there is to a DNA molecule? Is that right? No matter if you're talking about bacteria, which are prokaryotic, or eukaryotic, 4 domains. This is it. This is what this molecule looks like. And that's right. That's exactly right. Now, I want you to, I seem like I was forgetting something. Sometimes students have emailed me later, so I'm going to make sure I talk about this now, and I think it's a good question. You hear about, and y'all already know this, you hear about monomers or subunits that make up big polymers, right? And y'all all know, because it's been drilled into you, that cells are pretty much protein factories. Has that been drilled into you? If it hasn't, it should have been. And you know that proteins are a class of organic molecules that are made of what subunits? Did everybody know that? If you didn't, you should write it down. They're amino acids. Made of amino acids, they come up and get in line. Is that right? Is that right? So proteins are big macromolecules that are made of individual monomers called amino acids. And amino acids come together like cars on a train, and they line up and they make these big proteins, right? Is everybody good with that? So people ask me about, well, what is a nucleotide? I keep hearing nucleotide. What does that mean, a nucleotide? Nucleotides are the monomer units of nucleic acids. Just like amino acids are the monomer units of what? Proteins. Nucleotides are the monomer units of nucleic acid, of DNA, and of RNA. So to be a nucleotide, if we just look at this DNA molecule, let me tell you what a nucleotide is. It's going to be one or more phosphate groups, a sugar, and a base. So what I want you all to see here, to see if this is, I don't know how the back people could see a single thing, but this is telling you. So in nucleic acids, it's phosphate, a sugar, and a base. That makes up one nucleotide, one. So if we look at this DNA molecule, which is just a little piece of DNA, we said there's phosphate, sugar, phosphate, sugar, phosphate, sugar. But if we look, there's phosphate, sugar, and a base. Guanine, do you see that? And this is like connecting with phosphate, sugar, and a what? A base on the other side. Y'all see that? So this is the monomer. This is the monomer that makes up, and many of them come together to make up DNA. DNA has, depending on the species, has, like in humans, billions of bases. They're nucleotide bases. A phosphate, a sugar, and a base. Right? And when we think about the strand of DNA, we do know that it's double-stranded, that are phosphate sugar strands, with the nitrogenous bases as the rungs. And what we also know is something called the law of complementary base pairing, which I do want you to know, the law of complementary base pairing, which tells you that every time there's an adenine on one side, what will it base pair with? Same. And every time there's a guanine on one side, what will it base pair with? Cytosine. Everybody got that? Will there be any other pairing? No, this is the law of complementary base pairing. We know it base pairs like this, right? So when we think about this DNA molecule and how it's a twisted ladder, y'all see that? That's called a double helix. That it's this twisted ladder. We know that what's holding the rungs of this ladder together, this law of complementary base pairing, this chemical bond that's holding them together, is it a strong bond or a weak bond?
Strong.
It's an incredibly weak bond. It's the weakest of all bonds. It's the weakest. What's holding the adenine to the thymine and vice versa, and the cytosine to guanine, is this attraction. They're called hydrogen bonds. They're just attractions. And that suggests to you that this strand can readily do what? The strand can readily do what? Open up. It can readily open up. But I will tell you, it does readily do that, but it does take enzymes. And how would I know helicase is an enzyme? Because it ends in ASE. It does take these enzymes that are working to allow for it to do what it's doing as fast as it's doing it. These are some of the enzymes, and I would, as I said, I would have known they were enzymes by just their last names. I am not going to ask you to match them to their functions. If you ever take, some other classes, you would probably have to learn that. That's one of the things they would. I think that's whatever you look up to see what the functions are. But I would want some of their names tell you what they're doing, right? But obviously, we would realize that it's going to be important that the DNA can unzip. Is it unzipping right here? These are the two original strands. Do y'all see that? And are they unzipping? Are they? And it's going to take some enzymes to help to start the attraction of nucleotides to come in and set up. Can you all just look at this picture and just this image? It tells you that the original strands, after they have unzipped, the original strands will act as the blueprint for the new strands of nucleotides to come and get in line. Do y'all see that happening? So this is actually referred to as the law of semi-conservation. So before a cell can divide, the DNA has to make a copy of itself. Is that a hard thing to do? Not necessarily, because what's holding the strands together, a strong bond or a weak one? Pretty weak, so the strand will open up. The old strands will be the blueprint for the new strands to build on. And because of the law of complementary base pairing, as these two new strands are, these become the two new strands, if you will, as these strands are, and each of the two new strands has one old strand, that was its blueprint, law of semi-conservation. These two strands are identical to the first. Do you get that? Does anybody have any questions about that? Like, I know some of you had this in AMP1, but some of you I don't know. Do you have any questions about that? You feel good about that?
Re-listen to it.
If you read this, okay, fair enough.
Yeah.
So, okay. So thank you, Casey. The law of semi-conservation, instead of writing, y'all just look for a second. It just says you have a DNA molecule. It unwinds, it unzips. And the two old strands, original, are going to be conserved. And they're going to be the blueprint of where the new strands new nucleotides come in and lay down. So semi-conservation means that the old strands are conserved. They're conserved, aren't they? And they're going to be half of the new DNA strand. Half. Does that make sense? Now they're waiting frantically. All right, so that's fine. So this is how I want you to think about when you think about a cell needing to undergo mitosis. What is mitosis? Cell division. And is mitosis a type of cloning? Yes. Cloning simply means making a copy of yourself. When DNA unzips like this, using the old strands, conserving the old strands for the new nucleotides to lay down, building the two, aren't these going to be identical to what the first was? How do you know? Because of the law of, well, semi-conservation is the law of complementary base pairing. So if you can't see it on a large scale, let's look at it on one scale, right, Molly? So Molly, if we said adenine is right, let's say right here, this one's about to break. Adenine's on this side. What did you say is on this side? Thymine, and now it breaks. And a nucleotide, right where this adenine was here, what's going to lay down beside it? Thymine. And what's going to lay down beside this thymine on this side? Which is a copy. Right? Do you see what I mean? And that's doing, or do you see that? So that's like beautiful, isn't it? It really is. Really is. Okay. Now, does it take a lot of enzymes for this to be working? And enzymes are actually proteins. And proteins are actually built because of the DNA itself, right? So the cell has to keep the supply of those. Now, again, I'm not going to have you learn these enzymes, but I would think that it's important that you know that they're there and they're present. So this kind of shows you a picture too of this is how this is happening, and what would this cell be? Is probably written up there, but that has a circular chromosome. It would be bacteria, right? So how it's happening in bacteria. But in bacteria, we call a cell divided, cell division. What do we call that? Fission, right? So we said binary or prokaryotic fission, didn't we? That's how they divide. Is that a type of cloning? Do bacteria clone themselves? Yeah, they just clone themselves. They do, and really, it's kind of boring the way that they do. But yeah, so they just like clone themselves. Now, that's how DNA replicates itself. That's how DNA replicates itself. But now I want to review how DNA functions in a cell, because we said cells are all essentially what kind of factories? They're all essentially protein factories. That's what cells are. Now, cells can make other molecules as well, and they do. Cells make lipids. They do other things as well, and they can they can make a lot of what they need. to survive. The things that they can't make, that they must take in from the environment are called what? Essential. Yeah.
So when I sell nutrients, how does it like change the environment?
Okay, well, we're going to get to that. That's good you're asking. That's good you're wondering, though. It is, because we're about to get to that. So what I want you to know now is this idea about how DNA is the recipe book for all your proteins. I want you to understand how that happens. Because we get that the DNA molecule, this DNA molecule is, I'll get to a picture. We get that this DNA molecule here, this double helix, twisted double helix, somehow is holding the recipes for all of our proteins to be made. But what we need to know first is that actually recipes are really held on only one strand, on one side. It's not, a recipe isn't held on both sides. It's not like there's a recipe from here to here. The recipe is only on one strand, like from here to here. And the strand that holds the recipe, the side that holds the recipe, is called the sense strand. The other side of the DNA is referred to as the non-sense strand. But to get to a recipe, if it's in a closed molecule, what would we have to do? We would have to what? We would have to open it so that we could see the side that's holding. So guess what can happen? It can open. And did we say it's easy to open up right where the recipe is, or hard? Because it's what's being holding them together. The weakest of all bonds, hydrogen bonds. So with the use of enzymes, that recipe will readily open. And so do y'all see how it's open right there? And now we can read what we're reading, though, and see if this doesn't make sense. What we're reading are actually the sequence of what the sequence of those 4 bases are. And now I want to get this back to something else, because didn't you say that in the rungs there's only 4 bases, no matter if you're talking about E. coli, Mycobacterium leprae, Mycoplasma pneumoniae, us, a liver cell, a kidney cell, a neuron. Didn't you say that there's only 4 bases? What makes the difference in all the variation we see of all the traits is the difference in the sequence of those 4 bases. Look around at your people in this room. The reason you see different variations is because the sequence of the 4 bases are different among us. And what you see between species is that there's a different amount of chromosomes and that different sequence of those 4 bases. That's mind-blowing, that just the difference in the sequence and numbers of those 4 bases, that's mind-blowing, tells you the difference. It doesn't have the recipe for that particular protein that's being made. The sense strand has it. There are 2 steps getting from DNA to proteins. Those 2 steps you would need to remember are transcription and translation. Those 2 steps are called transcription, and the second step is called translation. Which one do y'all want to start with? The first one, which is transcription. Let's make this make sense. Guys, to transcribe something means to what? To transcribe it is to rewrite it, is to write it, isn't it? So, and certainly to write it, you know, you have to understand it. So let me tell you with transcription, I'm not sure I'm loving this picture. I like my little drawings better. Should I do it? All right, I'm doing my drawing for those people who hate my drawings, sorry. So if I had, you know, I've got 2 strands, but the strands are phosphate, sugar, boring, 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 boring. That doesn't change, right? So I know I've got 2 strands, but I'm only going to write the one because it's my sense strand. And I'm just going to make up a sequence here. So I'm going to make up a sequence of, I don't know, A, T, A, C, C, and maybe one more G, okay? All right, so that's my sequence, right? So, and I'm saying this is my sense strand. Transcription is the first step of getting from DNA to a protein. And what's going to be written is a molecule of another type of nucleic acid that's totally different. This nucleic acid that's going to be written is a nucleic acid that's actually called messenger RNA. And one nucleotide at a time will come up and get in line right here. One nucleotide at a time. It'll have a phosphate, a ribose for the sugar, and it will have a base. It will have a phosphate, a ribose. Did y'all say a nucleotide is a phosphate, a sugar, and a base? And they will come and get in line. Now look, RNA is a little different from the DNA molecule. RNA does not have thymine. RNA has instead uracil. So wherever there would have been a thymine, because on the complementary DNA strand that's open down here, isn't there a thymine right there? Isn't there, guys? But instead of a thymine, there's going to be a what? Uracil. Now, RNA does have adenine. RNA has adenine. And then there will be a phosphate and a sugar, a phosphate and a ribose, a phosphate and a ribose. And what would be right here? Well, it's, RNA doesn't have uracil. I know you do that. And then what would be right here? Guanine. And what would be right here? Guanine. And what would be right here? Cytosine. And how did y'all know that? Including that because of the law of complementary base pairing. So what you're saying is that you know that once the DNA opened up and the recipe was exposed, if you knew the sequence of the bases on the DNA, are you telling me you would know what the messenger RNA would look like as it's coming up and being written? And what was that step called, being written?
Transcription.
And are you going to know that?
Yes.
If that's what that step is called? Really?
You said it's a uracil?
Uracil instead of
Uracil instead of thymine.
No, I know, but like.
We just-
RNA has uracil. It's going to be in here in a second. URACIL. So uracil. But you know, y'all listening? RNA is also, messenger RNA is also single-stranded. DNA is what? It's just that the nonsense strand got out of the way for a minute. It's going to go back together as soon as that recipe is written. So transcription is happening on the DNA molecule. Transcription is producing a messenger RNA. Messenger RNA is a different nucleic acid from DNA. Messenger RNA is in that it is single-stranded. It doesn't have thymine, does it? In its place, it has what? Uracil. And it has a different sugar. What is the sugar difference? Ribose instead of what? Exactly. Are y'all good with that? You should. So you feel good? Because that's exactly right. That's right so far. You ready to keep going? Because so far I haven't said anything. I told y'all that we're about to learn about DNA, getting from DNA to proteins. I already told you that proteins are actually a sequence of what? Amino acids that have come to get in line. But I haven't said a single thing about amino acids coming up and getting in any line. So you know that can't be the end of the story. That can't be the end of the story. But are you going to first get that messenger RNA that's holding a message? You know what you could use instead of the word message? You could use what? Recipe for protein to be built. But we got to take that messenger RNA and we got to translate it. So this is what's this next step is translation. Fair enough? Mm-hmm. Fair enough? So at the end of translation, what will you have? So you will have a protein, and I want to show you how that is going to happen. And I actually think that this, okay, all right, well anyway, I'm going to write it because I think my notes are better. But then when you see the illustrations, they should make a little bit of sense, okay? Somebody tell me, just from what you had in your notes, since you don't, then you won't have to change it. Tell me what our messenger RNA looked like. Was it a U right here? It was a U, and what was right here? A, and what was right here? Just so you keep them the same in your notes. What'd you say? And another G, and a C. Is that what y'all had in your notes? Okay. So this messenger RNA, once it's made, will unlaunch from the sense strand. Is it hard for it to do that, or was the bond holding it kind of weak? The bond holding it was weak. It was just a hydrogen bond. So it unlaunches, and it's now going to go to this workbench that we know. This workbench, which is actually ribosomal RNA. So these molecules really have an affinity for each other, but this ribosomal RNA is the ribosome, the workbench, where this messenger RNA will come. You go with that? Everybody go with that? All right. Then what's going to happen? I think something's got to be translated. And what's got to be translated is this sequence from this RNA molecule is going to somehow have to be translated to a sequence of amino acids. What? Let's see how that happens. Before that happens, I need you to understand that a set of 3 bases, sorry that this other stuff's up here, this click to add me. A set of 3 bases. Is this 3 right here? A set of 3 bases on the messenger RNA is called a codon. Do you know why it's called a set of 3 is called a codon? Because they're coding for something. So they're going to be coding for something, right? And I'm going to tell you what they code for. They code for a little molecule, single-stranded molecule that's the anti-codon to it, that matches it. And only this y'all spell anti-codon right here. This is going to be only one particular sequence that could ever come up right here. And what would this sequence look like? This would be a what? An A. This would be a U, and this would be a A. Are you telling me that a AUU couldn't get right here? Or a UUA? Are you sure? Mm-hmm. Because this is an anticodon, and why do you know that they can't get, something else couldn't come up? Because of the law of what? The law of complementary base pairing. And this is called an anticodon because it is the opposite of the what? Codon. And we know it's very particular in which sequence can come. But this molecule is actually known as a transfer RNA, and it's called a transfer RNA. It should have been called a tiny transfer RNA, because is it tiny? It's got like 3 bases that are going to influence what's happening right here. Is that right? 3 bases, the anticodon. It's called transfer because guess what it's holding on to? An amino acid. It's holding on to an amino acid, bringing it to the workbench. Casey, you with me on this? It's bringing it to the workbench. It's bringing this amino acid up, and as it brings it to the workbench, it's going to let it go. It's going to unhook from it and let it go. And then another anticodon will come up and get right here and bring another amino acid to the workbench. And when those 2 amino acids get close enough together, guess what they're going to do? And when the next amino acid comes up and gets there because the transfer anticodon brought it, that's going to hook. And then that's going to hook. And now I've got a chain of amino acids. So guess what I just translated? I translated a messenger RNA to now at the workbench, I've translated a chain of amino acids that are happening, being brought up by these transfer RNAs that have anticodons that match up specifically with the codons on the messenger RNA because of the law of what? Yo, my amino acids can unhook. Okay, yeah, you get that.
So you said 3 bases is called a codon, and a codon makes an anticodon.
No, I didn't say a codon makes it. I said a codon is going to be specific in which it didn't make this anticodon. This anticodon is out there in the cells, but this is the only anticodon that can get right here. What would this anticodon be? What would this transfer RNA be? Oh, excuse me, I'm running over. So it would be C, what? C. Are you telling me no other one could get right here? No other? How about if a GGC wants to push its way in here, wants to get there, it can't get there because of the what? Law of complementary base pairing. It's not going to happen. And this one is going to be holding another amino acid, and they're going to bind. It's called a peptide bond. It's going to form between them. That's why whenever you hear about polypeptides, what do you know you're dealing with? A protein. You're dealing with a protein. I feel like if I'm yelling, it's going to work. What'd you say?
A peptide bond
Between the amino acids. Peptide bonds form between the amino acids. And now you've got this beaded chain of amino acids. Is that right? And what did we say proteins are? They're chains of amino acids that have come up and gotten in line very specifically. Is that right? Is that right?
Yes.
All right.
you hope. So,
Yeah, you hope. Or you're gonna
transfer
have
RNAs.
transfer RNAs are holding the anticodons that match specifically, because of the law of complementary base pairing, with the codons. And a codon is how many bases? A codon is 3 bases. A codon is 3 bases. And a codon calls up specifically an amino acid. Let me show you something, and hold on to your questions a second because it might be answered in the next little minute, okay? Let me see if I find a picture that I really like. Okay, maybe. Okay, maybe not. All right, let me show you this. If you look at this chart, if there are only 4 bases, but a codon is actually only 3 of the 4, and you know a base has something to do with the sequence of those 3 bases, you could actually, I could make you could actually tell me how many different sequences of 3 you could have. And you could start with adenine, 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 uracil, adenine, adenine. So you could do, but you'll take my word for it that there's 64 combinations. There's 64 combinations of codons. Do you see? Is UUU the same as UUC? No. And then there's UUA, UUG, and we could just keep going, right? But there's 64, and you would need to know there's 64 different codons. And you already told me that it's the codons that code up for the anticodon, which is the transfer RNA, that's bringing with it a what? An amino acid. So what you are looking at here, folks, and I never have anybody express the amount of awe that they should, but what you are looking at is the genetic code.
No.
Thank you, Jenna. You guys, that is a big deal, that this is the genetic code, and that we're just looking at it on a page. Do you know how much research it took to go into that to figure out the genetic code? Like crazy amount, like whole lifespans of people's careers went into this. So we should be in awe of this, but we know it. So now we can build on that, and this is how, medicine uses this all the time. Yes?
Why are there stops?
Okay, so hold on to some things, because I think I might get to some of these. I want you to know that this shows you, and we could count it, maybe you count them, but there are 64 of these, right? And what you're looking at is for each combination, exactly which amino acid is held on that anticodon, on that transfer RNA. So this codon is actually going to call up phenylalanine. This one, and this one does too. So this one has, phenylalanine has 2 different sequences that can code it up. But what we know is proteins, and you would have remembered this from your understanding proteins, they're made of chains of amino acids, and there's only 20 different amino acids. If you didn't remember that, you should. There's only 20 different amino acids. So intuitively, does that tell you that some of these codons code for the same thing? Because there's 64 codons, but there's only 20 amino acids. So some of them actually, and this is important to know for mutations that I'm going to get to, but this is important to know so that some of these code for these sequences of these codons code for the same amino acid. Now, so there's 20 amino acids. But wouldn't it make sense to you too that when a messenger RNA comes to the workbench, there has to be a place where the chain starts. A sequence that says, as it's getting there, start binding. And so if you look, I thought this is this AUG, it's the start sequence. So when the messenger RNA, on that messenger RNA, when AUG is read, is when the protein chain will start. The amino acid chain will start to make that protein. So you don't need to know AUG. I'm never going to ask you that, but it would make sense to you that there is a start and a stop, right? And actually, there are a couple of stops. There are a couple of sequences that will initiate stops. But that's just saying that that chain, y'all remember that we're protein factories, but proteins function the way they function because of their shape. Their shape is totally dictated on the sequence of the amino acids. If in a chain of 158 amino acids, there's only 20 different ones, but in a chain of amino acids that's got 158 different amino acids, if one amino acid is out of order, can it be lethal? Yes, it can be lethal. So is the sequence, the primary sequence, meaning the primary structure is the sequence of knowing which amino acid is in which position, is that important to know? Absolutely, because that's going to dictate how that secondary formation will occur. And when you hear about proteins denaturing and losing their function, which heat can do to them, or pH imbalances can do to them, or free radicals can do to them, then that's a big deal. Because if pH, all of those things, heat, free radicals, those things happen, then they can lose their shape. They lose their shape, and then they've lost their what? And you've already been hinted to that there are a lot of proteins that are making all this happen. What were those proteins? Those were proteins that were helping the replication of DNA, but also these steps, transcription and translation. It takes proteins making them happen. Do you want your proteins losing their shape and structure? No. Proteins don't typically have, they're not going to be made and live forever anyway. When I say live, are proteins alive? No, they're not. But they're not going to exist forever because they will eventually become denatured just because of metabolic. And then they have to be remade, don't they? So this is just a little bit of it. Now when we look, and let me get back to this. So when we look at this messenger RNA, you see the messenger RNA is this single-stranded molecule that has the bases in a particular sequence. Where did this sequence get dictated from? The DNA. Exactly. Because the DNA is the blueprint of all life, which means that it gave it transcribed this. A set of 3 will be the codon. And when you see the set of 3 for the codon, would you know which transfer RNA could fit right there? Would you know which one? Absolutely, because of the law of life. Each transfer RNA, which are much tinier than the others, they're going to have one end that you see the set of 3 is called the anticodon, right? You don't have to write that out because you already know the anticodons on the transfer RNA. Did you know that? But we also know that whatever is here is really going to dictate which amino acid can be held or hooked, right? And we know that by going and looking at the what? The genetic code, which is holding the genetic code, right? So how do you feel about this? So we've got this DNA sequence. We know the RNA codons are going to code up for particular anticodons, which are just transfer RNA, that are holding on to amino acids. But what would happen if you had, I'm trying to see if this is in there. Yeah, that's why they're called transfer RNA. They're transferring amino acids to the base. What would happen if you had a base sequence change on your DNA? What would happen if you had all of a sudden an inversion on the DNA? When is the DNA molecule most at risk for its bases to be changed? When it's doing what? When it's replicating itself. That's when DNA is most at risk for changes. Now, all living cells have built-in mechanisms to stop that from happening. But there are some things that do happen, and mutations do happen, and we're going to talk about different ones. I thought I'd get further today, but anyway, we'll talk about different ones in just a second. But they can be inversions, or there can be a deletion, or insertion. So those are all things that can happen. But if the DNA sequence changed, what could change?
The amino acid sequence, the protein.
Well, first what would change was the transcribed messenger RNA would change. And could that change which anticodons came up? And could that change the amino acid that came up? And if the amino acid changed, what could happen? The protein might be different. And if that's the case, can it be immediately lethal? Yes. Or could it just be variation? If you look at all the diversity you see in a species, most of those diversities happen because of single point mutations that occurred along the way that then became inherited. That's where variations come from. Every species that's been studied has a known rate of mutation, just spontaneous mutation that happens. It's really rare. It's really rare because the DNA molecule has built-in mechanisms to keep them from happening. So that's why it's so rare. But there is a spontaneous rate of mutation. And since we know that, what does that support? The theory of? Thank you. Evolution. That's exactly right, which just means change over time of traits, doesn't it? Oh, I spit on Brittany. That's right. It was right. It was just a little bit. Oh my God. Just a little bit.
That's why.
Why did you admit to it?
Because I thought y'all saw me. So, okay, so we have to get out of this room. But okay, do you have any other questions for me right now? This should have really been reviewed for my people. How about any other questions? Okay, so look guys, can we do it like a 10-minute break before lab? 10 minutes, okay? We are staining those microbes that you guys hopefully were growing the other day. You know it's Friday, isn't it? Are you doing it Friday? Are you doing it Friday too?